Hello, this is lecture 3. The outline of the lecture is introduction of T-cell responses, T-cell mediated immunity, definition and discovery of different Th subsets. In this slide, you can see the understanding the diversity of T-cell mediated response, immune responses. And you can see in this slide, CD4 helper T-cell play a crucial role in orchestrating the immune responses by coordinating the activities of other immune cells such as B-cell, T-cell, and macrophages. The slide describes is there a function of helper T cells carried out by a single type of cell, or there is distinct subset of helper T cells that perform different functions. This slide describes different types of CD4 helper T cells and their responses and functions. The slide describes the CD4 positive effector T cell subsets, their types and functions. The slide describes the naive CD4 T cell differentiated into different subsets based on types of microbes they are best able to combat, including intracellular and extracellular microbes as well as helminths. The slide describes the differentiation of Th subsets and factors involved. The slide describes the JAK-state cytokine signaling, a comprehensive overview of the mechanism, function, and regulation of JAKs and STATs in cytokine signaling pathway. The significance of shared JAKs in various cytokine pathways is the in-depth analysis of implication of therapeutic targeting and management of diseases. Th1 cells activate macrophages by producing cytokines which help in the immune response against intracellular pathogens. They also play a role in autoimmune diseases. The slide describes the role of human Th1 cells in a defense against intracellular microbes. The examples are below: Microbacteria or Salmonella. The slide describes the biological significance, mechanism, and regulatory factors involved in the differentiation and activation of Th17, including their role in immune defenses, autoimmunity, inflammation, and tissue homeostasis. Finally, the slide describes the role of STAT-C-dependent cytokine in Th17 cell differentiation, how IL-6, IL-23, and IL-21 contribute to